So finally, we have reached unit number 3. Let's start unit number 3, that is our next chapter in unit. Okay, so we'll be learning about chapter number 8 in our unit 3. So before that, let me just tell you an overview. In unit 3, you are going to learn about cell structure. All the chapters that you have, this 3 chapters: cell, the unit of life, biomolecules, and cell cycle and division, all are nothing but they talk about either the structure of the cell and they talk about function of the cell, what the cell is made up of. So biology is the study of living organisms, we know that, and detailed description of their life form and appearance. So in detail we are going to talk about the life forms and appearance that brought about the diversity. That's how there is a lot of diversity in living organisms, because of appearance. Based on appearance there are so many different type of organisms. Based on the form there are so many different type of organisms. So it is the cell theory that emphasize the unity of underlying this diversity of forms. So all living organisms are different, right? But the cell theory which spoke about that every organism is made up of cell, basic unit of every organism is cell. So every organism, whichever organism you talk about, ultimately it is made up of cell. So that is similarity, right? All organisms have something common, that is cell. Who told that? It was told by cell theory. So the cellular organization of all life forms is present in all the life forms. We have cellular organization. A description of cell structure and cell growth by division is being talked about in these further chapters. We'll be talking about that. The cell theory also created a sense of mystery around the living phenomena, that is physiological and behavioral processes. The mystery was the requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take physico-chemical approach and use cell-free systems to investigate. This approach enables us to describe the various processes in the molecular terms. The approach is established by analysis of living tissues, elements and compounds. It will tell us about different type of organic compounds that are present in living organisms. We are going to talk about that in biomolecules basically. Then we can ask question that what are these compounds doing inside the cell? What do they do inside the cell? This way we can talk about the physiological processes like digestion, excretion, memory, defense, recognition, all those things. We can talk about them based on what all compounds are present in our body. So we are going to answer the question, what is the molecular basis of all physiological processes? So physiologically, whatever processes we are doing, eating, digesting, whatever we are doing, what is the molecular basis? Based on every molecule, what is the basis of that? So we can explain abnormal processes that occur during any disease condition by using this. We can tell why the organism is getting diseased if you understand this. The physico-chemical approach to study and understand living organism is also called as reductionist biology. So we are understanding the physico-chemical approach to study and understand living organisms. So this is one living organism, right? So if I want to study this living organism, I am going to study the physiology of this living organism, and this physiology, to understand this physiology, I am using chemical approach. Chemical means chemically how this physiology, chemically how it is behaving, what it is made up of, structure, functions, all those things. We are breaking down into chemical. So that is why it is known as reductionist biology. We are reducing it into small chemicals. So chemistry basically. We are going to talk mainly about the chemistry and the physics. So using both we are trying to understand biology. That is going to be learnt in biomolecules. Now G.N. Ramachandran was an outstanding figure in the field of protein structure. He was the founder of Madras School of Conformational Analysis of Biopolymers. So yeah, this Madras School, in Madras School, he was the founder of that school where conformational analysis of biomolecules was done. So his discovery, triple helical structure of collagen Collagen is a protein, okay? So it is having triple helical structure. In Nature, in 1954, he spoke about that, and his analysis allowed confirmations of proteins through the use of Ramachandran plot rank. Among the most understanding contributions of structural biology, we are going to study about the structures of biology. That was done by G.N. Ramachandran. He lived from 1922 to 2001, okay? He was born in October 8, 1922, in a small town not much far from Cochin in southern coast of India. His father was a professor of mathematics at the local college and has considerable influence in, because of his father, because he was a professor, right, in mathematics at local college. Because of that, he got interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as top ranking student in BSc honors physics of University of Madras. He received PhD from Cambridge University in 1949. While Cambridge, Ramachandran met Linus Pauling and was deeply influenced by the publications on models of alpha helix and beta sheet. So, looking at Linus Pauling, he had written about alpha and beta sheet structures. Because of that, Ramachandran got influenced very much. That directed his attention to solving the structure of collagen. He wanted to find the structure of collagen. He passed away at the age of 78 on April 2001. So now let's start the chapter. The chapter is going to be very interesting. So let's discuss about this. That is cell, the unit of life. Okay, that is the basic unit. Every organism, the building block of every organism. How the buildings are made up of bricks. Like that, the unit from which our body is made up of, that is known as cell. Okay, so let's, without wasting any time, let's go deep into it now. So we see both living and non-living things, and we have wondered that what is it that makes an organism living and what is it that makes inanimate things. What is it that an inanimate thing does not have? What does a non-living thing not have, which living thing is having? What is special about living thing? So the answer is the presence of this basic unit, that is cell, which is present in all living organisms. So all the living organisms are composed of cell. Some are composed of single cell and some are composed of multiple cells, like us. They are called multicellular organisms, and single cell are called unicellular organisms. Now what is a cell? So unicellular organisms are capable of independent existence. Though they are unicellular, single cell, still they can live alone independently, and they can perform all the important functions of life. So anything less than a complete cell, whatever is there, that cannot live independently. So at least if you want to live, you need at least one cell. At least one cell is required to live independently. That is what unicellular organisms tell us. So the basic structural and functional unit of all life forms is the cell. So Anton von Leeuwenhoek, he was the first person who saw and described a live cell. He told about the live cell first time by looking at it. That is Anton von Leeuwenhoek. This is very important. So Robert Brown, later he discovered the nucleus of the cell. Inside the cell there is nucleus present within the cell. So that nucleus was described by Brown. Okay, which Brown? Robert Brown. And cell was described by Anton von Leeuwenhoek. So in later, when the microscope was discovered, an improvement was done in the microscope that created electron microscope and revealed all structural details of the cell. So cell theory, first time somebody described about the cell. So what does a theory mean? So theory means we are describing something, okay? Theory with lot of evidences, with lot of study, we describe something, how something is happening. We give big explanation to something, okay? We are explaining something in detail, something very big in detail, okay? That is called as theory. Okay, and whereas law means when you give equation to something, that is called law. equations when you are giving, that is law. But theory doesn't consist of equations. In theory we mainly explain big processes in detail. That is called theory, okay, by giving lot of explanation. So in 1838, basically in 1838, Matthias Schilden, so Matthias Schilden, a German botanist, he was German, and he examined a large number of plants and cells. See, so theory means not randomly somebody gave something. That is English word theory. So all of you need to understand the difference. In English, in when you talk about scientific theory, this is science, right? In science, a theory is something which has been observed, which has been seen large number of trials, large number of data, large number of evidences. All those make a theory. So theory is not something that is random. That is not scientific theory is not something that is random, okay. In English you have the word theory, which is normally we tell that can be any theory random, but in science theory is not anything random. So he observed lot of plants and animals and composed different kinds of cells which form tissues of the plants. So he observed that all the plants and animals are made up of different kinds of tissues. At around the same time, Schwann, okay, this was given by Schilden, right? Next, Schwann came. Schwann is a British zoologist. He studied different types of animal cells and reported that cells has thin outer layer, which today we know as plasma membrane. So though that time we didn't know about the plasma membrane, still he could describe it. So that was the speciality of this theory. He also concluded, based on studies on plant tissues, the presence of cell wall is unique characteristic of the plant cells. He told that if cell wall is present, that is present only in plant cells. That is what is special about plant. If this is human cell is just like this, but plant cell will have an extra cell wall around it. Okay, that is speciality of a plant cell, unique characteristic. So on the basis of Schwann proposed the hypothesis that bodies of animals and plants are composed of cells and products of cells. Schilden and Schwann together formulated the cell theory. Together both of them gave. So this cell theory is given by Schilden and Schwann together. So they gave this cell theory. This theory, however, did not explain as to how new cells were formed, because the function of this theory was what is the living organism made up of. It didn't explain how new cells were formed. Later, Rudolf Virchow, 1855, he first explained that cells divided and new cells were formed from pre-existing cells. This is known as omnis cellula e cellula. From pre-existing cells we get new cells. This is known as omnis cellula e cellula. He told that, okay. He modified the hypothesis. Who told that? This was told by Rudolf Virchow. How new cells are produced? From one pre-existing cell we get extra cells, okay. From that again we get extra cells. From pre-existing cell only new cells are produced. It was told by Rudolf Virchow. Schilden and Schwann, he modified the hypothesis of Schilden and Schwann, right, to give the cell theory a final shape. He gave the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and products of cells, which was told by Schilden and Schwann, and all cells arise from pre-existing cells. That is omnis cellula e cellula. That was given by Rudolf Virchow. So cell theory is consisting mainly of these 2 points. Now let's talk about overview of a cell. Basically, let's talk about little bit overview. So if you see the onion peel or human cheek cells under microscope, we can observe the cell structure. Okay, you can recollect the cell structure. The onion cell is a typical plant cell. Because why we study onion cell only? Because we can distinctly see the cell wall as its outer boundary, and just within it is the cell membrane. Within the cell wall it is having a cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. It doesn't have a cell wall. Outer layer is the cell membrane in the human cheek cell. Inside each cell is a dense membrane bound structure called nucleus. So outermost, if you talk about plant have outermost structure this thick structure what I'm drawing, this is the cell wall, and inside layer is called cell membrane, and inside that we can see a dark structure that is called nucleus. These are the main functions, main parts. But when you talk about human cell doesn't have cell wall, it just has cell membrane, and it has a nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material DNA. So this nucleus is made up of what? This nucleus is made up of chromosomes, which is nothing but it is made up of DNA. Cells that do not have membrane bound nuclei are called eukaryotic, whereas the cells that have not do not have sorry the cells which have membrane bound nucleus, so this nucleus is bound by a membrane, see it is having a membrane, so this is called eukaryotic cell because they have a membrane bound nucleus. But if this membrane was not present, just nucleus was present inside the cell, just randomly floating, this is called prokaryotic cell. That lack a membrane bound nucleus are called prokaryotic cells. In prokaryotic and eukaryotic cells, a semi fluid matrix called cytoplasm occupies the volume of the cell. So the volume of the cell is filled up. So this entire volume is filled up with this yellow color what I have drawn is cytoplasm. So entire cell is filled up with cytoplasm. The cytoplasm is the main arena of cellular activities in both plant and animal cells. That is the place where everything occurs. Various chemical reactions occur in it to keep the cell in living state. Besides the nucleus, the eukaryotic cells have other membrane bound distinct structures called organelles. They have endoplasmic reticulum, they have Golgi complex, they have lysosomes, they have mitochondria, they have microbodies, they have vacuoles. We learned about all these in detail later. The prokaryotic cells lack such membrane bound organelles. They don't have membrane bound organelles in the prokaryotic cells, whereas eukaryotic cells have membrane bound organelles. Ribosomes are non membrane bound organelles found in all cells. Because ribosomes are not membrane bound, so they are present everywhere. See, here also ribosomes will be present, here also ribosome will be present because they are not membrane bound. Both eukaryotic as well as prokaryotic, in them they are present. Within the cells, the ribosomes are found not only in the cytoplasm, but they are also found within the two organelles, chloroplast and mitochondria, and on rough endoplasmic reticulum also they are found. So animal cells contain another non-membrane bound organelle that is called centrosome, which helps in cell division. So centrosome, when the cell is dividing, right, when the cell is going to divide, the centrosome goes and aligns at the two corners. Okay, so the centrosome only helps the cell to divide into two pieces. So the centrosome is present in animal cells, that is non-membrane bound organelle, and it is called centrosome, which will help in cell division. Cells differ in greatly in size, shape and activities. For example, if you talk about mycoplasmas, they are smallest cell, only 0.3 micrometer in length, whereas bacteria can be 3 to 5 micrometers. So smallest cells are mycoplasmas, they are so small, whereas bacteria is 3 to 5 micrometer. The largest isolated cell, single cell, if you talk about, that is the egg of ostrich. It is the largest cell. Among multicellular organisms, human red blood cells are 7 micrometer in diameter. So this lengths you need to remember if you are preparing for NEET. That is the difference between NEET and normal board exams. In board exams, you don't have to remember very much in detail. If you understand the concepts, if you remember the names, if you remember basic things, it's sufficient. But in NEET, you should remember each and every point. So you need to watch this video multiple times if you're preparing for NEET exam. If you're preparing just for CBSE, watch the video once or twice and just read the textbook on your own, make your notes. All that is sufficient for CBSE. So nerve cells are some of the longest cells. Okay, so ostrich egg is the largest cell, and nerve cells are, they are not large, they are long, they are the longest cells. Okay, cells can vary in shape, they can be disc-like, they can be polygonal, they can be columnar, they can be cuboid, they can be thread-like, or they can be irregular. Also see nerve cell, I drew thread-like. They can be disc-like, cells can be like a disc, cells can be polygonal, cells can be columnar like a column, cells can be like a cuboid like a cube, they can be columnar like a column, they can be thread-like a thread, they can be irregular also, and they can be any polygonal shape. So these are different types of cells that you can see here. For example, this is our red blood cells. They are round and biconcave. Biconcave means from both the side they are concave. They are bent inward like this. They are bent inward like this both the side. So this is called biconcave. Whereas if you talk about white blood cells, they are amoeboid cells. Columnar epithelial cells, as you can see here, they are columnar. Epithelial cells means they form the boundary, right, outermost covering, that is called epithelium. Nerve cells are branched and long, see, they are long. And when you talk about tracheoid, they are elongated cells. Mesophyll cells are round and oval. So you can see different types of shapes are there. Now let's talk about prokaryotic cells. Prokaryotic cells are, they contain membrane bound organelles, as I prokaryotic cells lack membrane bound organelles, as I told you, because pro means first, carry on, karyotic. Okay, so karyotic means nucleus. When you talk about nucleus, that is karyotic. Pro means first, so nucleus is not true. Okay, nucleus is not, they don't have a true nucleus, so their nucleus doesn't have a boundary, that is they don't have a nuclear membrane, that is lacking here, that is known as prokaryotic cell. So prokaryotic cell are represented by bacteria, blue green algae, all the generally basic organisms will have prokaryotic cells. So pplo, pleuropneumonia like organisms, these are different examples. They are generally very small and they multiply very fast, other than eukaryotic cells. Compared to eukaryotic cells, they multiply very fast, so they may vary greatly in shape and size. Four basic shapes of bacteria are there: bacillus, which is rod shaped; coccus, which is spherical shaped; vibrio, that is comma shaped; and spirillum is spiral shaped. So if you talk about organization of prokaryotic cell, that is fundamentally similar, even though prokaryotes exhibit wide variety of shapes and function. If you talk about how the cells are organized in prokaryote, that is fundamentally it is similar. Okay, prokaryotic cell is similar. Even though there are variety of shapes and sizes, there are different shape and sizes of prokaryotic cell. Their functions are different, but still majority of them fundamentally they are similar. Basic structure is similar. Okay. So prokaryotic cells have a cell wall surrounding the cell membrane to protect them, except in mycoplasma. Mycoplasmas don't have no cell wall around them. The fluid matrix filling the cell is cytoplasm. There is no well defined nucleus, as I told you. The central genetic material is basically naked and not in nuclear membrane. They lack membrane bound organelles, right? So the genetic material is naked, it is not covered by anything. In addition to genomic DNA, the single chromosome or circular DNA, that is called genomic DNA. Okay, the single chromosome or circular DNA. In addition to that, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. Okay, so generally we have circular DNAs. Circular DNA means this is how the DNA will be, right? DNA is double helix. So DNA is present, single chromosome or circular DNA we have, that is known as genomic DNA, because they form the basic genetic structure of the organism. That is the main code of the organism. Along with that, they have some small DNAs outside, which is called plasmids. They have plasmids also along with that. So the plasmid DNA confers certain unique phenotypic characteristics to the bacteria. This plasmid DNA only gives certain different type of phenotypic characters, some extra features like resistance to antibiotics. Some bacteria are not killed by antibiotic. The resistance to antibiotic, even if you take medicine antibiotic, the bacteria will not die. Who decides that? That is because of this plasmid DNA, that is small circular DNA. In higher classes, you will learn that this plasmid DNA is used to monitor the bacterial transformation with foreign DNA. That you will be learning later. Nuclear membrane is found in eukaryotes. No organelles, like the ones in eukaryotes, are found in prokaryote cells except for ribosomes. So, except for ribosomes, no organelles like that is found in eukaryotes. Whatever organelles we have in eukaryotes, nothing is found in prokaryotes except for ribosomes. Prokaryotes have something differentiated form of cell membrane which is called mesosome. They have this mesosome cell membrane which is differentiated. The characteristic of prokaryotes. So see here, you can see this infoldings, right? They have this mesosome is having infoldings. See here, this membrane is having infoldings. That's why it is called mesosomes. Now let's talk about the cell envelope and its modification. So when you talk about the cell envelope of this, most prokaryotic cells, particularly bacteria, have chemically complex cell envelope. The cell envelope is very complex. That is outermost layer. The cell envelope consists of tightly bound three-layered structure. Outermost glycocalyx, followed by the cell wall, and then the plasma membrane. So these have three layers. Okay, outermost layer, inside we have another layer, then inside that is the plasma membrane. Okay, so outermost layer is made up of glycocalyx. It is because of that it is hard. Okay, then we have the cell wall inside that, and then finally is the inner structure which is the plasma membrane. This is how it will be present. So although each layer of the envelope performs separate functions, distinct functions, they act together as protective unit. All are protective unit. Their functions can be different. They have different function, but they protect the. So bacteria we can classify into two groups based on the differences in cell envelopes and in the manner in which they respond to staining process developed by Graham Wiss. So Graham Wiss was a person who discovered the staining process. Based on that, we can divide into gram positive and gram negative bacteria. Some bacteria, no, we have a gram stain. Gram stain is a color. Okay, so this gram stain is a kind of stain ink. So this some bacterias, if you add, if you take bacteria and if you add this gram stain to it, some bacteria will become colored. The cell wall becomes colored. That's why they are called gram positive bacteria, because they are positive. They show positive reaction to this gram stain. So they are called gram positive bacteria. But gram negative bacteria, they do not accept this gram stain. So they are called as gram negative bacteria. Okay, gram is the name of the stain. Glycocalyx differs in composition and thickness among different bacteria. The thickness of this glycocalyx, outermost layer obviously, is different in case of different bacteria. It can be loose sheath, that is called slime layer, very loosely it is present, while in others it can be thick and tough, that is called capsule. The cell wall determines the shape of the cell and provides strong structural support to prevent the bacterium from bursting or collapsing. The bacteria shouldn't burst or it shouldn't collapse, right? Burst means it shouldn't get bursted outward, and it shouldn't collapse inside, it shouldn't shrink also. This is the natural size, it shouldn't shrink. The plasma membrane is selectively permeable. The plasma membrane that you can see here, that is selectively permeable. It doesn't allow everything to go inside. Whatever is required by the bacteria, only that it will selectively it will allow to move. The membrane is similar structurally to that of eukaryotes, because plasma membrane is present in both prokaryotes and eukaryotes, so it is similar to eukaryote. The special membranous structure is mesosome, which is formed by extensions of plasma membrane into the cell. I showed you here. Mesosome, extensions of plasma membrane, it is going into the cell to form vesicles, tubules or lamella. They help in the cell wall formation. They help in DNA to replicate. They help in distribution of daughter cells. They also help in respiration, secretion process, to breathe from those pores. They can take respiration. They can secrete whatever things they have to secrete. Then they increase the surface area of plasma membrane, because surface area increases, right, because of folding. Then enzymatic content is there. So all those things these can do. Mesosomes in prokaryotes like cyanobacteria, There are membranous extensions into cytoplasm called chromatophores which contain pigments. So when they are containing pigment, these membranous extensions, when they contain certain pigments, they are called as chromatophore. Bacterial cells, they can be motile or they can be non-motile. If they are motile, they have thin filamentous extensions on the cell wall called flagella. Bacteria show range of numerous arrangements of the flagella. They have filament, hook and basal body. So filament, hook and basal body, these are the three components of the flagella. The filament is the longest portion that extends from the cell surface to the outside, correct? So this is the cell structure. We have longest structure. This is called our filament. Along with that, flagella, pili, fimbriae are surface structures of the bacteria that do not play a role in motility. So along with this flagella, they have pili or fimbriae on the bacteria which don't have any role in motility. They don't help in motility. Pili are elongated tubular structures that is made up of special protein. Fimbriae are, so pili are elongated structures, they are made up of simple protein, whereas fimbriae are small bristle like fibers which are sprouting out of the cell. When you talk about some bacteria, they are known to help to attach to the bacteria and rocks in stream and hosts. So fimbriae help the bacteria to stick to any rock or any host tissue because they are thread like, right? So they can attach easily. Now if you talk about ribosomes and inclusion body, in prokaryotes, ribosomes are associated with plasma membrane of the cell. They are around 15 nanometer to 20 nanometer in size, very tiny. Ribosomes are like this, small ribosomes will be present inside the cell. They are made up of two subunits, 50s and 30s. When present together, they form 70s. Okay, so these ribosomes are made up of two units, which is 50s and 30s subunit. When both join together, they form 70s subunit. Okay, in bacteria, ribosomes are the site of protein synthesis. Several ribosomes may attach to single mRNA to form chain called polyribosomes or polysome. So lot of single mRNAs, ribosomes can attach and form mRNA, and they form a chain called polyribosomes or polysome. Ribosomes of a polysome translate to mRNA into proteins. Inclusion bodies uh, Inclusion bodies, reserve material in prokaryotic cells, are stored in cytoplasm in the form of inclusion bodies. So food material, reserve material if you talk about, they are stored in the cytoplasm in the form of inclusion body. These are not bound by any membrane system. They lie free in the cytoplasm. Example, phosphate granules, cyanophycine granules, glycogen granules. So the food material inside the cell, when you talk about inside the cell, when the food material is present, these are known as inclusion bodies. Okay, along with that we have ribosomes. I told you ribosomes are protein. Ribosomes together form mRNA, lot of ribosomes. Okay, and they are made up of two subunits, 50s and 30s, both together form 70s. Gas vacuoles also can be present sometimes, which are found in blue, green and purple and green photosynthetic bacteria. In all those we get this. Now let's talk about eukaryotic cells. Eukaryotic cells are the cells that are present in humans, correct? And all protists except Monera, generally everybody has eukaryotic cell. We learned that. So in eukaryotic cell there is extensive compartmentalization of the cytoplasm through the presence of membrane bound organelles. So because membrane bound organelles means they are nothing like wall, right? So they divide the cell into different compartments. So that is why extensive compartmentalization. We can say that the cell is divided into various compartments because of membrane bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. They have a nuclear envelope. That's why they are called eukaryotic. U means true. Okay, wherever you see the word U in biology, it means true. True nucleus. The nucleus is true. It is well developed. It is having a proper boundary around it. Nuclear envelope. So eukaryotic cells have wide, complex locomotory and cytoskeletal structures. The genetic material is organized into chromosomes, obviously. That is what the genetic material is made up of. Next, all eukaryotic cells are not identical. Plant and animal cells differ in the former processes of cell walls, plastids, and large number of central vacuole which are absent in animal cells. So plant cells have cell wall, they have plastids. Plastids are green color. Okay, so human cells don't have any color, right? Green color. And large central vacuole, because plant cells store food, human cells don't have to store food. So because of that, these three things are not present in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. Animal cells have centriole for division, because animal cells, when they divide into two, they have some centriole structures, I told you. You can see the structure here for plant cell and animal cell. So down, the structure that you can see is the animal cell here, and this is our plant cell. Okay, so as you can see clearly here, plant cell is having our cell wall. Okay, plant cell has this cell wall. Then if you go inside, this is our plasma membrane. Then if you go see, nucleus also has a nuclear membrane. This middle portion is obviously our nucleus. Then around, this is called nucleolus, sorry, and this entire thing is called our nucleus has a membrane boundary. Then you can see Golgi apparatus. Then you can also see endoplasmic reticulum. You can see rough, if ribosome is attached, it is called rough endoplasmic reticulum. If there is no ribosome, it is called smooth endoplasmic reticulum. Golgi apparatus, you can see Golgi apparatus is something like this. Then you can see mitochondria here. Then you can see the food chloroplast here, which is containing green pigment, that is our chloroplast. Then you can see big food vacuole here, this big vacuole here, which is not present in animal cell. You can see here, or otherwise all the parts are same in animal cell. This is the nucleus, this is the this is nucleolus, this is our nucleus, this is our ribosomes present on this endoplasmic reticulum, so it becomes rough endoplasmic reticulum. You can see our mitochondria, you can see the Golgi apparatus, you can see these nuclear membrane, nucleolus, nucleus, all the parts can be seen here. And centriole, I told you, centriole is like this kind of structure, two centrioles you can see here. Let's talk about the cell membrane now. So cell membrane, you know, cell membrane is the, after the advent of electron microscope, the cell membrane could be seen, the detailed structure of the cell membrane. So chemical studies on the cell membrane, especially human red blood cells, helped the scientists to deduce the possible structure of cell membrane. Plasma membrane. These studies showed that cell membrane is mainly made up of lipids and proteins. The major lipids and phospholipids that are arranged in a bilayer. Also, lipids are arranged within the membrane with its polar head towards the outer sides and hydrophobic tails towards the inner part. So, the hydrophobic tail, which you can see, that is present towards the inner part. This is hydrophilic head. Whereas if you see this tails, small tails are present over here. See, this is going to be the structure. Okay, cell wall is this cell membrane is made up of phospholipid. So this is our head, and uh, so this hydrophobic tails, these tails are not water loving. Hydrophobic means they don't love water, so they are facing inside. Hydrophobic tail, whereas this head is hydrophilic, it loves water. Hydrophilic, and two hydrophobic tails are arranged together because they are separated. And here we have our head again. So this is how the structure of the cell membrane is. So this ensures that the non-polar tail, the tail is non-polar, right? It is made up of saturated hydrocarbons. It is protected. Saturated means single bond, there is no double bond. Okay, so lot of carbon, this tail is made up of carbon, 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 carbon. Lot of carbons are attached together, single. It is protected from aqueous environment. Along with phospholipids, membrane also contains cholesterol, some amount of cholesterol. Now, biochemical investigation later revealed that cell membranes also possess a protein and carbohydrate. They have protein also and carbohydrate also. The ratio of protein and lipid varies considerably in different cells. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipid. So 52% it is made up of protein. You can see here it is having peripheral proteins. It has some sugars attached. So this is, but mainly this is our cell membrane. It has some proteins, integral proteins. In between it has proteins. It has peripheral protein. Peripheral protein means the protein which is outside. Integral protein means which is part of the cell membrane. In between the cell membranes there are some proteins, and you can see some amount of cholesterols are present here. So this is the structure of our cell membrane basically. Depending on the case of extraction, membrane proteins can be classified into integral and peripheral protein. Peripheral proteins lie on the outside surface, as I told you, and integral proteins are partially they are present inside or completely they are buried in the membrane. So an improved model of the structure was called fluid mosaic model. It was given by Singer and Nicholson. So according to quasi-fluid nature of the lipid enables us to lateral movement of proteins and overall bilayer. The ability to move the membrane is measured as its fluidity, because the membrane can move, so it is referred to as fluid. The fluid nature of the membrane is also important for the point of view of like cell growth formation, because cell has to grow, it has to be fluid, right? It has to be able to expand, and intercellular junctions, secretion, endocytosis, cell division, etc. So when you talk about one of the most important functions of plasma membrane, it is the transport of molecules across it. The membrane is selectively permeable. It allows only some molecules to pass through it. So there is passive and active transport. Passive transport means directly, because of diffusion, some particles can go across the cell membrane directly. That is called passive transport without using any energy. But active transport requires energy, which is called ATP. ATP is the energy of the cell. Example is sodium potassium pump. So sodium potassium pump is a kind of for transmission of sodium and potassium here and there. So if I want to transfer Na plus ions from this side to that side, I require energy in the form of ATP, energy currency. This is known as our active transport. Neutral solutes move across the membrane by the process of simple diffusion along with concentration gradient from higher concentration to lower concentration. For example, here I have more concentration of something, here I have less concentration of something. So from more side to less side they will easily flow. This is known as passive transport. They can directly move. And movement of water, when it is happening, when these molecules are nothing but water, if it is made up of water, then it is called diffusion. That's it. It is very simple. So through the non-polar bilayer, as the polar molecules cannot pass through the non-polar bilayer, this require carrier protein through the membrane to facilitate transport. So non-polar molecules cannot move from this side to that side, right? So they require a carrier protein. Here in between there will be a protein which is going to carry this from this side to that side. That is known as our carrier protein. From lower to higher concentration. So if from something is going from lower to higher concentration, such transport requires energy, and that is why it is called as active transport. Now let's talk about cell wall. Cell wall is obviously, I told you, to protect. So a non-living rigid structure is called the cell wall. It is outer covering of the plasma membrane of fungi and plants. Cell wall gives shape to the cell and protects the cell from mechanical damage and infection. It also helps in cell to cell interaction and provides barrier to undesirable macromolecules. It is providing barrier for the undesirable big molecules. So big molecules which are not required, that cannot pass through the cell wall, so it is called as this macromolecules, so will not be passing through that, through the cell wall. Algae have a cell wall made of cellulose, galactins, mannans and minerals like calcium carbonate. These are the things. The algae cell wall is made up of cellulose, galactins, mannans and different minerals like calcium carbonate here. So other plant cells contain cellulose, cellulose, pectins, proteins, all the cell wall of a young plant. The primary cell wall is capable of growth and gradually diminishes as it matures, and the secondary wall is formed on the inner towards the membrane of the cell. Middle lamina is a layer mainly of calcium pectate which holds different glues, different neighboring cells together. The cell wall of the middle lamina can be traversed by plasmodesmata which connects the cytoplasm of the neighboring cells. So from one cell to other cell, if I need to have a connection, from this cell to other cell, if I need to have a connection, how it will be present? In the form of plasmodesmata, that is connection between two cytoplasms of neighboring cells. Is it given here in the diagram? Plasmodesmata. See here, you can clearly see here plasmodesmata is given here. These are connections. From one cell to other cell you will have some connection, pores. Two cells are attached like a pipe. Two cytoplasms can be attached together, that is called plasmodesmata connections. Next we are going to talk about endomembrane system. Inside the cell we have some membranous system, so that is known as endomembrane system. It is made up of endoplasmic reticulum is an endomembrane system, Golgi apparatus is an endomembrane system, lysosomes, vacuoles, all these are endomembrane system. Inside the system we have, so endoplasmic reticulum, you can study the here names only are given basically, and functions of mitochondria, chloroplast, all these are not coordinated with above components, and then they are not considered as part of endomembrane system, that is mentioned here. Electron microscope studies of the eukaryotic cells, they tell about the presence of network of reticulum tubular structures scattered in the cytoplasm. This is called endoplasmic reticulum. Endoplasmic reticulum divides the intercellular space into two distinct compartments. One is luminal, that is inside the endoplasmic reticulum side what is present, that is called luminal, and extraluminal, that is cytoplasmic components which are present outside. Next, endoplasmic reticulum shows ribosomes attached to it. If ribosomes are attached to it, it is called as rough endoplasmic reticulum. If ribosomes are not attached to it, it is called smooth endoplasmic reticulum. Rough endoplasmic reticulum is observed in cells actively involved in protein synthesis, because ribosomes are nothing but protein, and protein synthesis and secretion, who does? Rough endoplasmic reticulum. They are extensive and continuous with the outer membrane of the nucleus, whereas smooth endoplasmic reticulum, smooth means you can remember lipid is smooth, so the animal cells lipid like steroidal hormones are synthesized in the smooth endoplasmic reticulum. Now let's talk about Golgi. Since Camillo Golgi discovered it, it is called as Golgi complex. It is densely stained reticular structures. Stained means when you stain the cell, it gets color, right? So it is, this part is densely stained. Near the nucleus it is present. It was later named as Golgi bodies after Golgi, Camillo Golgi. They consist of many flat disc-shaped sacs, cisternae. You can see here it is having disc shaped sacs here. These are known as cisternae. Many disc shaped sacs are present. 0.5 to 1 micrometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi cells. Different number. Cisternae are concentrically arranged near the nucleus, distinct convexes or forming face, and the concave trans or the maturing phase. Trans is the maturing phase. Cis is the forming phase. Okay, the phase that is getting formed, that is called cis phase. The cis and trans phases of organelle are entirely different but interconnected. The Golgi apparatus primarily performs the function of packaging the materials together. It does the work of packing. Okay, this word becomes very important here. To be delivered either through the intracellular targets and secreted outside the cell. Materials to be packed in the form of vesicles from ER, endoplasmic reticulum, gives material. Okay, so first the materials are produced from the DNA or RNA that codes for the. Next we have our chromosomes or nucleus. Okay, so materials will come from nucleus basically. If I want to tell, instead of DNA I can call it nucleus, then it goes to our endoplasmic reticulum. Then it is having ribosomes around it that can form smooth rough endoplasmic. Then it goes to Golgi apparatus for packing. So materials are made, then it is packed by the Golgi apparatus. Move towards the maturing phase from cis phase, that is not maturing phase. It will go to trans phase, that is maturing phase. A number of proteins synthesized by the ribosomes and the endoplasmic reticulum are modified in the cisternae, that is proteins, nothing but packed, right? So they are synthesized by the ribosomes on endoplasmic reticulum. Ribosomes work is to synthesize protein, and they are modified by the Golgi apparatus before they are released into the transphase. Golgi apparatus is the important site for formation of glycoproteins and glycolipids. Next, let's talk about lysosomes. Lysosomes are nothing but membrane-bound vesicular structures formed by a process of packing of Golgi apparatus. So, after Golgi apparatus, after packing, what is formed? Lysosomes are formed because they are packed with membrane-bound vesicular structures. The isolated lysosomal vesicles have been found to be rich in almost all type of hydrolytic enzymes. They contain hydrolysis. Hydrolysis means hydrolytic enzyme. Lase means breaking. Hydro, water. So lipases, proteases, carbohydrates, all these different type of hydrolytic enzymes are present. They are active at high acidic pH. When the pH is acidic, these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Next, let's talk about the vacuoles. Vacuoles are membrane bound space found in the cytoplasm. It contains water, sap, excretory product, and materials that are not useful for the cell is present in the vacuole. Whatever is not useful. Next, vacuoles can occupy 90% of the volume of the cell. I showed you. Then the vacuole bound by a single membrane is called tonoplast. This is very important. Tono means single membrane. So in plants, the tonoplast facilitates the transport of number of ions and materials against the concentration gradients into the vacuole. Hence, the concentration is significantly higher in the vacuole than in the cytoplasm. In cytoplasm, the concentration of ions is very high. Amoeba, the contractile vacuole, is important for osmoregulation and excretion. In many cells, as protists, food vacuoles are formed by engulfing food particles. Amoeba is nothing but it engulfs the food material. This is food material. It will come around this and it will engulf it and it will take it inside it. Next, mitochondria. Mitochondria single is mitochondrion. Unless specifically stained, they are not easily visible under the microscope. If you would stain them normally, they are not visible. We should specifically stain them with mitochondria stain which is used particularly for mitochondria. The number of mitochondria per cell is very variable. It depends on the physiological activity, what the cell is doing. If the cell requires lot of energy, there will be more mitochondria. If it requires less energy, the cell will require less mitochondria because they are powerhouse of the cell, right? So in terms of shape and size also, considerable degree of variability is observed. When you talk about sausage shape, cylindrical shape, different shapes are present. 0.2 to 1 micrometer diameter they have. Average is 0.5 micrometer and length is 1 to 4.1 micrometer. Each mitochondria is having double membrane bound structure with outer membrane and inner membrane dividing the lumen distinctly into two aqueous components. You can see here it is having an outer membrane and it has an inner membrane. Then inside it is having matrix. Then intermembrane space it is having between two membrane. Then it has cristae, that's foldings. The foldings that you can see are called cristae. So same thing, whatever I told about the structure is mentioned here. The inner compartment is filled with dense homogeneous substance called the matrix. Then the outer forms limiting boundary of the organelle. Then cristae is mentioned, that is foldings, infoldings. The cristae increase the surface area. The two membranes have their own specific enzymes. Mitochondrial function. Mitochondria are sites of aerobic respiration, that is to take oxygen and they do respiration. They produce cellular energy in the form of ATP. Hence they are called powerhouse of the cell because they produce ATP. ATP full form is adenosine triphosphate. That is energy currency of the cell. Who is producing that? Mitochondria. So RNA molecules, ribosome, 70S, the components required for the synthesis of proteins. The mitochondria divide by fission. By fission, the mitochondria will divide. So it's a big chapter, little bit. So plastids are found in different parts of the cells, euglenoids, then chloroplasts. Different colored pigments are called plastids. You have chlorophylls, you have carotenoid pigments that is made up of carotene. Then you have elaioplast, amyloplast store carbohydrates, elaioplast store fats, oils, all those. Aleuroplast store proteins. So different plastids are there which store different compounds. Elaioplast oil and fat, amyloplast carbohydrates, and aleuroplast proteins. Then there is given different colors also here. Carotenoids pigments contain these different color. The part gives a plant yellow, orange or red color. So different colors it gives these plastids. Then you can see ribbon shaped organelles with length and width is mentioned here. Chlamydomonas has one per cell. So all those are given, like mitochondria, chloroplast are also double membrane bound. You can see here they have double membrane bound, outer membrane and inner membrane they are having. Algae has how many, that is given here. So thylakoids are number of organized sacs. You can see here different sacs like it is stored. So that is known as our thylakoid. Disc-like structures. Flattened sacs are arranged. That is called thylakoid. They are present in stoma. Thylakoids are arranged in stacks like pile of coin, which is called grana. Singular granum, it is made up of granum or the intergranular thylakoids. In addition, there are different membrane bound storage lamina. All these you can read. 70s and 80s ribosomes are present. Next, ribosome is nothing but, I told you, these are dense particles by George Pallet. He discovered it mainly. And ribonucleic acid, they are composed of that, and proteins, and are not surrounded by any membrane. Eukaryotic ribosomes are 80s, whereas prokaryotic ribosomes are 70s. Next, when you talk about eukaryotic ribosomes, they are made up of 60s and 40s, two subunits, whereas this 70s is made up of 50 plus 30s. So they have a large subunit and a small subunit. Both are joined together. So large subunit is 50 or small is 30, 60 or 40. So S here means Swedberg's unit, which stands for sedimentation coefficient. It is indirectly measure of density and size. Both 70s and 80s ribosomes are composed of two subunit only. Next, if you talk about cytoskeleton, that is nothing but the skeletal structure basically here. So elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, intermediate filaments present in cytoplasm, effectively referred to as cytoskeleton. So cytoskeleton of the cell is involved in major function like mechanical support, motility, maintenance of the shape of the skull, just like skeleton. Cilia and flagella, all of you know, you can read what is cilia and flagella. Cilia is cilium, singular is cilium, flagella is they are hair like outgrowths present on the cell. They are nothing but movement and all. So cilia, flagella, you can see. Okay. Centrosomes and centrioles also you can read, that is organelle consisting of two cylindrical structures, amorphous poly centriolar materials. They are made up of. Centrioles are perpendicular to each other. They have organization like a cartwheel. You can see here cartwheel like a kind of structure. Nucleus you can study next. Nucleus is made up of, you know about nucleus, all those things are only given, just read it. The remaining chapter you can just read it. 2-3 part points are left over. Nucleus is very important, and our chromosomes are very important. So chromosomes have short and long length. Top portion is known as satellite. Some will have satellite. This middle construction is called a centromere. It is having short arm, long arm. Based on position of centromere, it can be divided into short arm, long arm, all those things. So they are named. Okay, different names are given based on. Metacentric means if it is present in the middle, submetacentric if it is present little below, then acrocentric if it is present almost at the end, then telocentric if the centromere is present at the end only. So these are different positions based on that. Satellite is a small fragment appearance it gives. Non-staining constructions which are present here, it gives a small fragment like appearance, which is called satellite. Microbodies are, they are minute vesicles. Microbodies, they contain various enzymes and they are present in both plants and animals. So thank you very much. Do read the NCRT once so that you understand, because I don't have lot of time to explain everything in detail, but I have covered major part. You can just read the remaining things on your own, and if there are any doubts, please make sure you ask in the comment section below.